Hello, and welcome to the Carl Zeiss webinar, Sigma and Merlin 3View, Fast and Convenient Imaging for Tissue Samples in the FE-SEM. My name is Maya Everett. I work in the marketing group for Carl Zeiss in the U.S. I would like to thank everyone who is joining us today, both our participants and our panelists. First, some general information. During this webinar, you will watch approximately 45 minutes of presentation, followed by 15 minutes of Q&A. Audio questions are disabled. However, please submit questions at any time via the chat window in the GoToWebinar menu shown on the right-hand side. Questions will be selected from the chat window after the presentation concludes and will be answered by our panelists. The recorded webinar will be available on our YouTube channel. To review our agenda, Joel Mancuso from Gatan will start by discussing 3View and serial blockface imaging in the FE-SEM. He will be followed by Dr. Martin Edelman from Carl Zeiss, who will review the 3View with Zeiss FE-SEMs. And then Tom Deering of UCSD will present applications for the 3View. With that, I'd like to hand it over to Joel.
Thank you, Maya, and thank you everybody for attending. And just a quick overview of what 3View is, and it's known in the literature as serial blockface scanning electron microscopy, or the acronym SBEM. And it really refers to the modality of how we're using the SEM. We're using the SEM completely different. We're using it in a way that we're going to take images that resemble more of a TEM image than an SEM image. So just to kind of describe the acronym, the serial refers to, we've actually taken a ultramicrotome and placed it in the chamber of an electron microscope. And what that enables us to do is to image a surface of the sample, and then the microtome will shave off a portion of the sample, expose a new surface, and we image that next surface. And we do that over and over again. So that kind of refers to the serial modality. We're going to take a series of 2D images to create a three-dimensional data set. Blockface is more of it's a complete change of how we're acquiring the image. In TEM, we cut a thin section, usually 70 to 90 nanometers thick, and we collect a projected image. With this microtome in the 3View system, we use the microtome to shave off a surface, so we're no longer imaging the actual section that's been cut off, but we're imaging the new surface that's been revealed. So just a little bit about traditional images that you can collect with an SEM and an TEM. And on the left-hand side, we have kind of a classic red blood cell image. And on the right-hand side, we have an image of a synapse. TEM has been predominantly the more useful tool for life science and biologists, because you can see what's going on internally, what's going on with the mitochondria, what's going on with the synaptic vesicles, where are they distributed. Where with the traditional scanning electron micrograph, you can only really get an idea of what's going on the outside, or you can get a general idea of the morphology. Is there different in shape between the cells? If we look at a serial blockface image, again, this image was taken in SEM, but it resembles very much of what we're used to seeing when we collect an image with a TEM. So this is just a quick movie kind of giving you a general feel of what the instrument looks like. We started out as zooming out, showing an SEM, and SEMs are a little bit easier and more straightforward to use than a TEM. We replaced the door of the SEM with the 3View. So the 3View has its own door, and on that door is mounted a microtome. That microtome is equipped with a diamond knife, which shaves the surface. And here we've just cut the surface of the sample. We're going to raster the beam over the surface of the sample. So the imaging modality is a little bit different. We're no longer taking a projected image. We're going to collect an image by rastering the beam point by point across our sample and collecting a backscattered electron image. Once we've collected the image, we bring the knife back over to the sample, shave off, in this example, 15 nanometers, and collect another image. This whole process is completely automated and really expands the capabilities and the volumes of tissue that you can image. So I'm going to briefly talk the two real important points and why I believe 3View is just an exciting product and how it's going to expand, bring back electron microscopy in the biological sciences. And we'll start with automated imaging. I believe that traditional life science or biomedical electron microscopy, or TEM, is like, the analogy is a manned lunar mission. The results are exceptional. They're the highest resolution images you can get in the life science when you compare it to light microscopy. Unfortunately, it's difficult to collect these images. You need a highly trained individual. You need that individual to sit in a dark room for hours or days, depending on what you're imaging and how many images you'd like to collect. I have a lot of experience sitting in dark rooms, and often and even in the old days when you use film, I would shoot an entire box of film, developed a film, and then I'd realize that half the images I'd taken weren't of use, or maybe there was slightly stigmated, or maybe it was slightly out of focus. And overall, endurance is a big problem. You can't take hundreds of microns of images through a 3D data set. So if we keep with the space analogy, the way that NASA solved this is they created a robot. And this robot does all the imaging. So instead of actually sitting in the room and pointing out, instead of going to Mars and going and picking up each rock and taking a picture of it and coming back home, we just tell the robot, okay, take a picture of all these rocks. And then when you're done taking a picture of rocks, go to the horizon and take a picture of the horizon. And while the robot's doing that, I go to lunch and I come back and I can start scanning through the pictures immediately. And I'm looking through the data. I'm not looking at the actual rock, or in our instance, I'm not looking at the actual section on the microscope. And I think this is exactly what 3View does. Because the imaging is completely automated, we can set this machine up to run overnight, and it will cut through 50 microns of tissue. When I come back in the morning, I can take my cup of coffee, I sit in front of the computer, and I can scroll through the images one by one. I can sit with my cup of coffee and casually go through the data instead of being in a confined room and hoping that my shot is perfect and absolutely focused. The system does that automatically for us. And I think this will give us great capabilities to image large data sets. The other really important point is that 3View simplifies electron microscopy. I believe that the most difficult part of doing electron microscopy is not the microscope. It's actually cutting thin sections, ultra-thin sections, and collecting them on a grid. It's sitting in front of the microtome. That's the barrier of entry. That's the real art form. I can train anyone to use an electron microscope. I can show them what buttons to push. There's no real... In focusing, you could argue, is an art form, but sitting in front of this ultramicrotome, it's very difficult to train someone, and I often had the most difficult training people on the ultramicrotome when compared to the TM. So I believe that that's the barrier of entry, and 3View successfully removes that barrier of entry. I use this movie as a story to kind of elucidate this idea. This was a movie taken by a group at the University of Helsinki, and they hired a software engineer to create automated segmentation algorithms, which you'll see shortly. Some of the cells will pop up in different colors, and that was done automatically. You didn't have to go and trace the cells. So this gentleman, his name is Ilya Belovich, he's the software guy. He decided to sit in on the 3View training, so just to learn how the images are acquired, how the machine works, and maybe he could write a more robust algorithm. I saw him 6 months later at the Society for Cell Biology meeting in Denver, and he showed me this movie. And I said, oh, this is fantastic, look at that, and we're talking about the segmentation, and then he casually says, well, I collected the data too. I actually set up the microscope and ran it, and I'm actually I'm in charge of the microscope. I'm collecting all the data and annotating all the data. So within a 6-month time frame, there was an individual who had never sat in front of an electron microscope, who didn't even have a background in microscopy. He was a software engineer. And within 6 months, he was creating beautiful data sets, and arguably at the time I'd say this is probably one of the most beautiful 3View data sets we have as a robust application example. So it really illustrates that this is a very straightforward and approachable technique. So why do we do EM? And I think EM has always been one of the imaging techniques that's a fallback. A lot of my experience with working with individual researchers is they would submit a paper, and the paper would get conditionally accepted if they could prove X, Y, and Z with a couple of electron microscopy images. And no one in the lab knew how to do electron microscopy, so they would come visit us. The real reason we do this is high resolution. We need high resolution and structural data. And this is really the only technique to do that. What's unique is we're now combining those high resolution ability with 3D ease. Before, if you would like to take serial images to create a movie like we're seeing now, you would have to cut, let's say these are 100 sections. That would take, maybe you can do that in the afternoon. And then it's going to take you 3 days, maybe 2-3 days to image. And then you have to correct all the distortions and put it together. And so this data set would take a week if you did it traditionally by TEM. This was done in less than 2 hours on a 3D ultramicrotome. So there's real power in automating the process and reducing the amount of time to data. So once we have these serial images, what are people doing with them? And I think the most obvious thing is we want to understand what's going on in 3D space. We want to make movies. We want to make models of a particular cell. And this is just a small volume. This is 25 microns by 25 microns, 500 serial sections, or serial images in this case. This data set took about 8 to 10 hours to collect, so just overnight. But the real difficult part is actually creating that 3D model. And people are working on automated segmentation algorithms, but in this particular case, we went out and retraced this, this portion of the dendrite by hand and to create this beautiful 3D model. I mentioned that, and I want to harp on these points that 3D is really simple and 3D is completely automated. And what can you do when things are simple and completely automated? And I think this is a great data set illustrating that you're really starting to break barriers in electron microscopy, where this data set, so it was about 6,000 sections, so that's over 300 microns in Z. It just wouldn't be possible to do it by hand. And if some poor graduate student was working on their PhD and they were able to do this, it would take many, many years. This data set took 9 days to collect, and the system is robust enough and it's stable enough to be continually collecting images for weeks on end. Winfried Denk, who's the kind of inventor of this imaging modality, has had data sets run up to 3 weeks. And so it's very, very stable. So it's a beautiful advantage of this technique. And the title of this slide is Beyond TEM, and I want to illustrate that not only are we collecting these really massive volumes that are just not possible by the traditional technique of serial sectioning, but we're also cutting thinner. Because we're not collecting sections, we can cut the surface and shave the surface off much thinner than what you would normally do for cutting a section for TEM. And this is an example of cutting at 15 nanometers thick. And what we're seeing in this front panel is the image, the collected image, so this is the XY plane, and below we're looking at the YZ, and to the right we're looking at the XZ. And you can see that in the XZ and YZ we can still see individual vesicles. So these are 15 nanometer sections. You couldn't cut that thin using a TEM, so that's why I illustrate really pushing beyond what the TEM is capable of. I'm just going to wrap up with 2 more slides, and these are stories from customers, application examples from customers. This is from the Cleveland Clinic, Graham Kidd and Bruce Trapp. They've had a system for a while, and they are interested in the central nervous system. And so we look at the back panel here. These are 4 3D data sets. Each image is 8,000 pixels by 8,000 pixels, and a couple, I think about 500 images deep. What they were able to do is, if we go directly below, you can see this axon. This is about a 500 micron length of an axon, and it's in 3D because they have the complete serial image data set. In the red box, we're looking at the real resolution of that axon. So it's a high-resolution data set. Graham stood up in front of a group of people and said, you know, I'm not sure I'm the first person to look at this length of an axon in 3 dimensions, but I am sure that I'm the first person to look at 50 of them. So what they did was they pulled out 50 individual axons from this data set, and they were able to ask and answer very simple questions. They were just interested in where are the mitochondria? Are they located near the nodes? Because that's the real action. That's where you would think that the most energy would be needed along the axonal length at the nodes of Ranvier, or are they just randomly distributed? And this was kind of a question that plagued the literature. And they were able to collect enough data to generate statistical analysis of the data set, and they realized that they're kind of randomly distributed. They are randomly distributed. They are not located at the nodes. And so they kind of were able to really solidify that because of the amount of axons, 50, and the amount of mitochondria that they counted in each axon. And that's just something you wouldn't be able to do by serial sectioning. Another question they ask, and they're interested in a central nervous system disease, and they have a knockout of this particular model in a mouse, and so they compare the wild type to the knockout. And they were just, again, looking at what do the mitochondria look like between the wild type and the knockout. And they looked at 2 different time points, 1 month and 6 months. And when you just look at volume, there's not a huge difference between the wild type and the knockout. The wild type's in green and the knockout's in orange. The blue is a control from a different region of the brain. The real story here is the length. When you compare the wild type to the knockout mouse, the knockout, the mitochondria never really extend into this cigar shape that the wild type or the healthy mouse has. And again, they were able to collect enough data to generate 3D models of mitochondria and to do statistics on the models. And that's something that you don't have enough resolution in a light microscope, and you just don't have the real estate taking serial images in the TEM. And this was a data set that they collected overnight, and they were able to generate hundreds of models, and so it had enough numbers to generate meaningful statistics. And I think that's what's been largely missing when you look at 3-dimensional electron microscopy. People make these pretty models, but pretty models don't get us published. We need hard numbers. And so this is a real step forward for just 3D EM in general, but especially for 3View, because it really illustrates the power of the technique, showing how easy it is. You can collect this data overnight, and then the power of how to generate meaningful statistics from these data sets. So that's about all I have, and at this point in time, I'd like to hand over the presentation to Martin Endelman. Thank you.
Well, thank you, Joe, for this nice explanation of the system itself, of the technique of 3View. I will go on with telling you a little bit about the count size FE-SEMs and why we think that these are especially suited for supporting this application with these 3View applications. I have to tell a little bit about the technology inside our SEM chambers, inside our the column inside, which is the GEMINP technology, very famous actually. And there are several issues in this technology which are optimal supporting the 3View application. We have, as a emitter cathode, we have a field emitter which is necessary to have the stability. And as you remember, you have seen examples from Joe which runs for 9 days or longer. So it is absolutely necessary to have a very stable system. And also this field emitting cathode is necessary for having highest resolution. Then the GEMINI allows to have probe currents up to 300 nanoampere, and that is really a lot. Normally you never use that in life sciences. But you can use very high currents, and that means in that application that you are able to scan very fast without losing resolution. And so it is absolutely an issue. It's a big difference for you if you run a sample overnight or if you run it for weeks or for months. So to speed up the imaging and the scanning of the sample is an absolutely important thing. Then we have a very special feature inside the GEMINI technology. This is true for the GEMINI 2 column, which is the improved version. It is the double condenser, and that allows you to freely choose the current what you want. There are not apertures inside that. You are completely free to choose the current what you want, and at each current you have always the optimum of resolution. That's very special in this design. And also very special is the beam booster. This is especially for having high stability for low voltage operation. As I will explain very shortly, it is absolutely necessary to do all these imaging at low voltages. For example, 1 kV or 1 to 3 kV low voltages. And in that GEMINI design, the beam boosters has the option or provides a very high stability by putting the whole column onto an 8 kV level. That means even if you are using very low kV as a probe current at the end of the sample, inside the column, the electrons are having 8 kVs or more. And that brings a very high stability inside the electron column. And of course, because they are very fast inside the column, they have to be decelerated at the end of the column. So we have an in-lens deceleration of electrons. This is a very stable design which allows for very long-term experiments. Then we have two very special detectors inside this GEMINI column. I will not go in detail on that. The reason for that is that we have also a detector from Gatan together with the 3-view system. But anyway, these detectors are included in the column, and they allow to separate between secondary electrons and backscattered electrons very nicely. And very important again is the special final lens of the column, which is very good suited for large area scanning because of very low magnetic fields at the sample surface. It is possible to image large areas without any aberrations or distortions. I will show you examples shortly. Altogether, this is the GEMINI design, which is the basic design of all our FE-SEMs. This brings us to the advantages of using this column for the 3-view. This column is very special supporting the low voltage operation, and that is very important because only with low voltage you achieve that the electron beam is penetrating not too much into the sample. So the penetration depth is very low, and that means you are getting only the signal from the top surface of the sample, which you cut away afterwards. If you have too high currents, you will go too deep into the sample, and you will get signal out of the sample from a depth what you don't want, because you want to have the signal later with a later slice. The further important thing for low current is, for low voltage, is that you can operate in high vacuum mode, because with low voltage you can achieve that the sample is not charged, and in Otherwise, you would have to use a variable pressure system, which is also possible. But here you can use a high vacuum mode. So all these are important factors for doing low voltage operation, which is optimal, supported by the Gemini design. And the Gemini lens, as I explained, is very good, suitable for a large field of use, up to 32K times 24K pixel resolution. And that is important for these large volume imaging, because if you think you would have to do everything with, for example, in this image here, 8K times 8K, you would have to stitch a lot images to each other. And this is time consuming and produces failures. And so very often we have customers which are absolutely satisfied to do the 32K times 24K imaging, and that's enough for them, and they don't have to stitch at all. We have several systems which are supporting the 3-view operation. There's the Sigma VP and the Merlin Compact VP. These are mid-range systems in our portfolio, and they are offering the Gemini 1 technology. And they have also a special issue, that is the high current mode. That simply doubles the probe current without losing too much resolution. And that means you can again scan faster your sample. You can double the scan speed with almost the same resolution, and that gives you double the speed, and so you have in half the time your results. And these are systems which are able to have variable pressure, which you can use for charge compensation if you want. If you want to have very high speeds, you will go with the Merlin, which is the highest current, which has the highest current spot size ratio in the SEM market. With this very high current which it can drive, it is able to image 10 times faster than comparable systems. It has the best signal-to-noise ratio, and so it is optimal if you want to do very large volumes. If you have really great and high data volumes, I think it's the best system in the market. And as I explained, it has these double condenser designs, which allows you an aperture-independent probe current adjustment. Let me show finally, in my part, 2 examples with the Sigma 3-view, because Tom, after my talk, will more focus on the Merlin. These are examples from London, from Peter Munro's lab. He's in the University College London. This is, for example, a dendritic cell. It was almost 1,000 slices. He reconstructed 72 of them here in this example. A pixel resolution of 5K times 5K, so it's not too much. Pixel size 12 nanometers. It's just to show you it is very easy, and it's a matter of very short time. I don't know the actual time here. Very easy to do imaging, 3D imaging of whole cells, and then after that you can segment the parts you want, as you see here, the cell nucleus, endoplasmatic reticulum, mitochondria, and all that stuff. Still a nice example which can be done with many other samples in the same way. And here we have another example, again, thousands of images. It's just to show you that you get very, very fast large volumes. This is not a very high resolution example here, because the microtome was set to remove 100 nanometer, and as you heard from Joel, it would be able to go down to 50 nanometer. But anyway, if you have here a data set which has overnight produced a 100 times 100 times 100 micrometer data set. So very large volume overnight, and it gives you still a lot of information in this example. Well, and with that, I would like to hand over to Tom, who will go a little bit further into much deeper applications and also with Merlin applications.
Thanks, Martin. My name is Tom Dierink, and thank you all for joining the webinar. And time is short, so I'll get right to it. I just want to start out by saying that in all the years that I've been doing EM here at the National Center, I really have never seen a technology that is so potentially transformative to EM as I think serial block-based imaging is. As Joel said, it's essentially an automated 3D EM robot. And I believe one that will revolutionize not only the way that we collect EM data, but how we view it as well. And I'll show examples of that. We've been using this technology now for a number of years, most recently with two primary platforms, a Zeiss Sigma VP and a Zeiss Merlin. In fact, ours was the first Merlin to be equipped with a Gatan 3 view, and I have to say, Mark Ellisman, the founder and director of NICMER, was taking a bit of a risk in that all previous 3 views had been installed on variable pressure SEMs to deal with specimen charging, and our Merlin is high vacuum only. The two main reasons he took the chance was that we believed that we could mitigate specimen charging by how we prepared the samples, and that the Merlin could deliver the combination of high current and very small spot size at very low voltages with the stability that we require to operate the system nearly continuously, and fortunately Mark was correct. Now I'd like to go over very quickly specimen preparation for 3 view, since when we introduced our improvements for block face imaging at the Microscopy and Microanalysis meeting 2 years ago, there were so few people doing serial block face at the time, we simply provided a link to the protocol rather than publishing a paper on it. Probably a mistake, but because it really does make a huge difference in the results that you get. Now we've been preparing samples for high voltage EM for quite a while, and we're pretty familiar with heavy metal staining of tissue. In fact, we published a paper over 30 years ago on metal impregnation of nervous tissue so that it becomes so metallized, it becomes conductive enough to be imaged by SEM without needing any coating. So for the 3 view, what we did is combine existing metal stains into what can be described as an extreme or enhanced heavy metal staining protocol, to say the least. It uses conventional glutaraldehyde fixation, followed by ferrocyanide reduced osmium tetroxide post-fixation. This is then followed by a treatment with thiocarbohydrazide. Thiocarbohydrazide is a bifunctional osmium binding ligand that binds to the osmium in the tissue, leaving a remaining free osmium binding site. You then wash away the unbound thiocarbohydrazide and follow with a second round of osmium. And this binds to the unreacted sites on the thiocarbohydrazide in the tissue and almost doubles the osmium content. You then do a prolonged staining with uranyl acetate, then en-bloc staining with Walton's lead aspartate, which is a lead stain that was introduced almost 30 years ago by Judy Walton. And then you dehydrate and embed in epoxy resin. Preferably, we use a very hard version of Durkapan ACM resin. So let's see, that's osmium, iron, more osmium, uranium, lead. It's a lot of metal staining. Now, since the specimens become so metallized and partially conductive, we want to take advantage of that. So we also modified the way in which the specimens are mounted. We remove the excess epoxy from the backside of the block to expose the tissue and mount it with conductive epoxy to the pin. We then trim the block to expose tissue on all 4 sides. And finally, we sputter a layer of gold palladium over the entire specimen. This way, there's a continuous direct path from the tissue to ground, and many types of specimens, as I said, become conductive enough so that variable pressure imaging is not necessarily required, and you can do block face imaging at high vacuum. And the results are pretty impressive. Under identical imaging conditions, if you compare the image of the conventionally stained sample on the left with the enhanced staining on the right, and as I said, same microscope conditions, the image quality is dramatically improved to the point where you can even easily see synaptic vesicles. And as I said, this protocol was introduced at M&M in 2010, and the link to it is right here at the bottom, and it is still active. It's been adapted for use in everything from cultured cells to worms to the first synthetic bacteria that Craig Venter invented to most major tissues. So it's also being used in many groups around the world. Even people doing dual beam and electron tomography are using it because the specimen contrast is so great. Now, if I can offer you one piece of advice, if you want to try your samples for review, you should use a protocol like this on your samples first of all, because it will make a big difference in the type of results that you can get. And this brings us to our high vacuum Merlin. This instrument was meant to really push block face imaging to the limit using state-of-the-art electron optics so that we would not be instrument limited in our efforts to make better specimens to maximize resolution. The Merlin has a number of key attributes that are essential for the highest performance block face imaging. These include the ability to maintain high current and in a very small spot at very low accelerating voltages, as I said before, allowing for both fast and high resolution block face imaging at 2 kV and below. And we can scan 32K fields at high resolution and do so quickly. But perhaps the most critical thing is the stability that the system has demonstrated at low accelerating voltages at these over extended periods of time because of the design of the Gemini column. The 3-view system itself is flawlessly integrated into the Merlin. We can see the new door and the stage containing the miniature robotic ultramicrotome here, the light. The ultramicrotome really is a fine piece of engineering design, if I may say so myself. And from the very start, the images were very impressive. This is a block face image of mouse cerebellum, imaged with a Merlin at 1.5 kV and at high vacuum. We can see this large Purkinje cell neuron, and if we zoom up on this boxed region, which you can see here, we can see the excellent detail that is resolved. This is a portion of the Golgi apparatus that we see down here, and we can actually see individual nuclear pores up here in the nuclear envelope. Very nice. Now, the resolution that you can get with a Merlin in block face images is so good that it can be difficult to tell the difference between a block face image and one produced by thin section TEM. Here are two very interesting images, so look closely. We cut a 60 nanometer thick ultra thin section from a block of cerebellum with a conventional ultramicrotome and imaged it by TEM. We then imaged the corresponding region on the block face by backscatter SEM. It's a little hard at first to tell which is which. But I think if we look here, we can see that the SEM image is the one on the left, acquired with a Merlin at 1.9 kV, and the standard TEM image of a section is, over here on the right is at 80 kV. And I think that there's really probably no doubt that the TE image looks better, but the block face image is actually pretty darn good. Now, another way we can tell which one is which is that the ultra-thin section invariably contains, as we can see here, various degrees of distortion and compression. This is how the TEM image looked when it came off of the microscope and before digital dewarping. Now, you can try to minimize this section distortion in serial section reconstructions, but it requires a great deal of attention to detail and effort, and this is something we just don't have to deal with using serial block face imaging. Now, how do you extract 3D information from 3-view image stacks? Of course, one way is to use manual segmentation, which can give very spectacular results. The reconstruction that you see here on the right is part of a project with George Spyro at West Virginia University. He's using it to study circuit development in the auditory pathway. And we have been providing his team with literally terabytes of robotically acquired 32K 3-view data from the Merlin. And they have been producing some absolutely stunning reconstructions. I just saw an animation of the latest series of whole cell reconstructions yesterday, and I asked George if I could show them here. They're just absolutely spectacular. But he wants to debut them next week at the neuroscience meeting, so if you want to see them, you're going to have to hunt down either George or Mark directly. Now, this manual segmentation process can be very tedious and labor-intensive, but fortunately there is a serious effort to develop auto-segmentation algorithms to streamline the process. Not surprisingly, really, to me, you can learn quite a bit just by looking at the image stacks without doing any segmentation or sophisticated image processing. As Joel said, instead of one person sitting in a darkroom in front of an electron microscope looking at a few sections, you sit at your computer or in a group in front of a monitor and browse through hundreds or thousands of wide-field image planes in 3D. It's like exploring your sample with Google Earth. And once you try it, you'll wonder why you ever did it the old-fashioned way. I think it really will change the way we not only acquire EM data, but the way we view EM data. So now time is short, and I want to end my talk by talking about something that I think is particularly exciting to me, and I want to thank Zeiss for giving me the time to talk about it, and that is our efforts here to create GFP-type genetic labels for 3View. Now this is a real challenge to label proteins in a bulk block of tissue without having to resort to all sorts of things like pore fixation and using membrane-disrupting detergents, as you have to do for immuno-EM, just to get superficial labeling, let alone uniform labeling over dozens of microns. But we believe we have a way. So we are designing specialized molecular genetic probes for EM that we build into the target protein at the genetic level. And then we can not only use this for serial blockface imaging, but also for general TEM protein labeling as well. We don't build the probe. We just design it and deliver a set of blueprints to the cell and get it to make the probe fuse to the target protein itself. We then use this fusion protein to make an EM visible reaction product using diaminobenzidine. So right now, there are, well, let me see, 1 second. So right now, there are 2 main types of genetic tags that we are developing for serial blockface. The first is Mini-SOG, which you see on the left. Mini-SOG stands for Mini Singlet Oxygen Generator, and is created with Nobel laureate Roger Chen. Mini-SOG is the first fluorescent protein engineered specifically for EM protein labeling, and I think it is totally revolutionary. The other that you see on the right is APEX, developed in the laboratory of Alice Ting at MIT. Alice is a former student of Roger's and a brilliant neuroscientist in her own right. The design of APEX is based on the plant protein ascorbate peroxidase, and is a small, is highly active in all cellular components. And I think it's going to be a blockbuster tool in the EM community, mainly because it is so darn easy to use and overcomes so many of the obstacles that we have had to just accept. Now, either of the tags can be cloned in very much like GFP, and using them is very straightforward, especially APEX. In cells or tissues that are expressing the recombinant protein, you fix the sample with conventional 2% buffer glutaraldehyde. Then in the case of Mini-SOG, you eliminate the sample with blue light in a solution of diaminobenzidine and oxygen, and the Mini-SOG photochemically creates a reaction product for you. Or in the case of cells that are expressing apex fusion proteins, you simply incubate the sample in a solution of DAB and hydrogen peroxide for a few minutes. You then postfix with osmium tetroxide and take it through the rest of the 3View protocol. If you're preparing the sample for conventional TEM, it is exactly one additional step in an otherwise completely conventional EM protocol. No compromises in fixation, no need for permeabilizing detergents, and you get high-quality 3D EM labeling. Now, I apologize that I'm very limited in what I can show right now in tissue, as APEX is very new. The paper describing it doesn't even come out in Nature Biotechnology for another 2 weeks. And there is a robust but still ongoing activity to incorporate Mini-SOG probes into a number of different animal models. But I'd like to show you 2 quick examples, if I can, in cultured cells. This is Mini-SOG used to visualize the adenoviral protein E4-ORF3 in the nucleus. On the left, we see a transmitted light image of cells before photooxidation. You can see the Mini-SOG fluorescence image by confocal here in green. And then here on the right, after DAB photoconversion. And then here, the image plane on the far right from the Merlin. It forms this very intricate intranuclear web. And here, this is an animation. moving from the top of the cell monolayer to the bottom, plane by plane, and it looks a little bit like confocal imaging stacks. But the nice thing is that we can use a simple intensity projection, as we're going to see here, to reveal a 3D distribution of this protein. So a huge advantage of this type of label with a 3D system is that just like confocal microscopy, you can use simple intensity projections and no laborious segmentation is required to get out your information. And here very quickly is an example of Apex fused to the chromatin organizing protein histone 2B, transmitted light image, and then here is an image from the Merlin. And I think this type of results is very, very promising. And I think I'm going to stop here so that we have time for questions. And again, I want to thank Zeiss for the opportunity to talk about these new probes, even though their application to serial blockface imaging in tissue is still very early. But I know they realize their potential to bring GFP-like labeling to serial blockface and want to keep their customers at the forefront of this technology. So as you can imagine, I think these probes will be a big boost to serial blockface imaging, just as the combination of 3View and the Sigma or Merlin field emission SEMs have been such a big boost to serial blockface imaging. And of course, I want to acknowledge our excellent collaborators, Mark Ellisman, the director of NICMER, and especially Roger Chen and Alice Ting for the work on the probe development. And so, thank you very much.
Okay, thank you, Tom. That was great. At this point, I'd like to hand it over to Martin for a Q&A session. I think we have about 10 minutes left. Martin?
Yes. Okay, let's have a look. So you still have the opportunity to fill in some questions in the questions fields, so we can answer that. We have got a few questions. One is for Tom, actually. The question is, what is the typical duration of a 3View experiment in your lab?
It varies, of course, by the type of specimen. The monolayers go very quickly because, as I said, we're operating at very fast dwell times. So you can go through a whole cell monolayers at 20, 30, 40 nanometer sections in the course of just a few hours. The longest run we've done on the Merlin is about 10 days, a little bit less, continuously operating, where we generated about 5 terabytes of imaging data. And that was to traverse a very large field of brain tissue.
Mm-hmm. Then a question I would say, Joel, this is something you can answer. It is that I've read that there are 2 versions of 3View. I think it is about 3View and 3View XP. Could you comment on this? When do I choose which version of 3View?
Yeah, no problem. So the 3View 2 comes in two flavors, the XP and then just the standard 3View 2. With the 3View 2 is we took a bunch of the automated features and we changed the stage from an automated click and point type stage to a manual micrometer stage. So the stages have to be manually moved by hand. The other important note for the 3View 2 is that the cutting thickness specification is 30 nanometers. For the XP, you get all the bells and whistles. You get the point and click automated motors. It's the cutting specification is 15 nanometers. And with the click and point automation, you get a couple other features. You can do montage where you can do a 4x4 or even a 10x10 image montage to stitch images together. We have something we call multiple ROI, where we take a very low mag image and then we draw several regions of interest on that image, and we take one slice and go to each of the ROIs, acquire an image, and then cut again. So you can acquire from different regions per cut. So the main difference is the stages and the cutting thickness.
Okay. Then if you have questions about the slice, I think it's still for you, Joel, about the slice thickness. What is the reproducibility of the slice thickness? How do you measure slice thickness?
We measure each system as they leave the factory, and we have a eddy current jig that measures the steps of how we move the sample up in Z. We measure at 15 nanometers for the XP system, and we have a couple different measurements. One measurement gives us about a standard deviation at just over a nanometer, and the motor encoder report gives us a standard deviation of 0.6 nanometers per step. So we are quite confident that when we say we cut 15 nanometers, we are cutting 15. We're not cutting 10 in one cut and 20 the next cut and 18 the next cut. And we are cutting 15 nanometers each cut.
Okay. Next question is here from Cynthia. I want to know how long takes normally the whole process from the beginning to obtain a 3D image. I assume it is about the whole process from sample preparation until getting the first image in the SBEM, running through the whole process.
Maybe Tom, that's a great question for you, since
That's
you're
Tom,
more of a user and practitioner.
Sure, sure. So the processing of the tissues or cells for EM is, there are more steps than conventional TM, but it's very straightforward. And generally the protocol can be completed in just 2 or 3 days. And then the samples, as you know, for EM are good for forever. And then mounting the specimen just takes a little bit of practice, but it's not very difficult. And then setting up the system is absolutely easy. And as Joel said, there is not a big barrier to people using the system without being very experienced electron microscopists. You can train someone very quickly to get on the system, set it up, and start acquiring data. And that's one of the most important and I think impressive things about it is that it's the robot that does all the heavy lifting. The operator is just kind of sitting by with a big smile on their face.
Yeah, John, you just can go on with the next question. This is, can traditional immuno-EM be used in 3D view?
Yeah, so this is a problem. We've been working on trying to improve protein labeling for EM for a long time. This has been a project that we've been working with Roger Chen for many years. And you can, but as you're aware, in order to be able to get antibodies in for labeling of tissue, oftentimes you have to use very compromised chemical fixation, and you have to use detergents such as Triton or saponin to get your labeling in even just a little way. Now, if you can use the techniques for conventional TEM, 3D EM, then those same samples can be used for 3D. But I think you'll find that it's almost impossible using these traditional techniques to get the labeling in over dozens and dozens of microns with well-preserved material and without having to use these disruptive detergents. So this is why we're directing things towards these new probes, this new way of labeling tissue. But yes, the short answer is, in limited fashion, you can use immunolabeled specimens.
The next question, I think, is for Joel again. Do we need to change the diamond knife from time to time?
Well, if Joel doesn't want to answer that, I can answer it.
Joel, still there? Okay, Tom, perhaps you can say something about that.
Sure, sure, because we've been operating these systems now, and as I said, they run nearly continuously every day, 24 hours, and they were just stopped between runs. And the ultramicrotome is very different, and I didn't know this at the start, than a conventional ultramicrotome because you're not cutting a section. And the number of cuts that you can get out of this diamond is much greater than you can get with a traditional diamond knife. I haven't done precise calculations, but I think it's orders of magnitude longer lifetime than a conventional diamond knife. And so this is one thing we're very impressed with, is the ability to cut and cut and cut without having to worry about the diamond knife edge.
I have here an additional question. This is in the same direction. Are there other consumables?
That's a That's a very good question. The pins that you mount the sample on, we consider consumable. The backscatter detector, because it's so sensitive, in the beginning we thought that we were going to treat it like a consumable item, but we rarely see problems with those in the field. But, you know, Tom's had a system for 3 years now, and we haven't replaced his detector. We haven't replaced his diamond knife. Probably the only thing Tom needs is the pins to mount the samples onto.
Yeah, that's correct, Joel. We're actually, the Merlin is behaving, and the Sigma, excellently, and the backscatter detectors have had no problems whatsoever with continuous operation. And in fact, the Merlin and Sigma have behaved so well. The Merlin has 15,000 hours on the field emission source and is showing no signs of degraded performance, so it's very nice.
Tom, a next question again for you, I think. Do you have any ideas to adapt the improved heavy metal staining for serial blockface imaging for freeze substitution?
Yes, this is actually something that I think the community would like, and we are working very diligently on to bring this technology to high-pressure frozen material. And there are some challenges, but we are making progress, and we do believe we will be able to show something, I think, probably at the next microscopy and microanalysis meeting next summer. We'll be able to show it publicly. But yes, there's a lot of effort in this direction.
Just to follow up on Tom's question, you know, Tom and Mark Ellisman, they were, you know, early adopters of this technology, and in the last year we've seen a huge increase of the number of systems that have been installed in the field. And I always tell people once they get their system that there are 2 areas that I think will be the most exciting, the next steps, the next improvements for this type of technology, because we've always borrowed from the TEM. Right, for this prep, even Tom's protocol of the heavy metal stain, it's an old high voltage TEM protocol. That's where the idea came from. And I believe that we're going to start seeing serial blockface tech, you know, specimen preparation, like new ideas. And I think the 2 places, like, again, is the staining, and that's one area. But the next is the resin, and creating resins that are more beam tolerant. The more tolerant the resin is, the longer you can dwell the beam, or the more dose you can put on your sample per pixel, and your signal to noise will increase, and your resolution can actually increase as well. So I think that there's the 2 real exciting areas is the resin and the sample preparation concerning high pressure freezing. So I believe there are many groups working on this. One group in Australia, Richard Webb, he recently got a 3-view system, and he's a big believer in high pressure freezing, and one of the first things he wants to do is get a high pressure freeze or high HPF protocol that will be compatible with 3-view.
Okay, we don't have any more questions, I think, and we also are at the end of our time. So Maya, I would hand over to you again.
Oh, perfect. Thank you. And thank you to, I just want to take the opportunity again to thank everyone for joining us and to thank our panelists for putting together a nice presentation. We welcome any additional questions or comments to our email address, micro@zeiss.com. All inquiries will be responded to within 1 to 2 business days. And again, thank you to everyone for a great webinar.
Thank you.
Thank you.
Thank you.